Now, what happens in the contractile cells? How does contraction happen is the big question, right? So this represents the inside cytoplasm of a contractile cell. So, and we're going to go through kind of what happens here. So the first thing that happens up here with number one is at the surface of these cells, we have some sodium channels, all right? So there are sodium channels here that will open and allow sodium to come in. And that causes the action potential to spread across the surface. What opens those sodium channels is these positive charge that is coming through the gap junctions that were delivered from the SA node. And that positive charge causes the resting membrane potential to reach threshold of -40. And then these sodium channels start opening all along the surface of the cardiomyocyte. And then sodium is going to flow in all along the surface. And as that positive charge dives down into a T-tubule, the sodium channels are replaced with voltage-gated L-type calcium channels. And so sodium channels up here initially cause the rising phase of the action potential. And then these L-type calcium channels finally open and stay open for a long time and cause what we're going to call the plateauing of the action potential. So the action potential of a cardiomyocyte, let me draw a little scale here, which I'll show you again later, is going to start off with rest, at rest, right? So we'll start off at rest, and then sodium channels are going to be stimulated to open, correct? And the sodium channels open, and then they are going to close, and then these calcium channels open for a long period of time, and then they close more slowly, like this. And so basically it's sodium first. These are called fast sodium channels. They open first and they're at the surface. And then we got these L-type calcium channels that open for a long period of time. And they deliver calcium in here. So sodium and calcium are coming in. And that's much different than the autorhythmic cells, which it was just calcium based, wasn't it, that caused the action potential. Now, I know you had some funny current sodium channels, but there are no channels associated with the action potential, the complete depolarization of the membrane. That is just L-type calcium. Here you got sodium and calcium. But when calcium flows in here in the T-tubule, the T-tubules, remember, are associated with the SR on either side of the T-tubule, deep in the myoplasm or the cytoplasm of our cardiomyocytes. That's going to open these L-type channels, this depolarization by sodium, and that will then open these RYR channels, which are called ryanodine receptors. So ryanodine receptor or channel. And again, it's going to be a calcium channel. All right. And so it's going to open in response to this depolarization. And then you're going to get calcium flooding the cytoplasm. This causes what's called a calcium spark. And then as more calcium is released and more SR is involved, you get this huge burst of calcium, which is called the calcium signal. And that calcium then bathes the myofibrils, which are made up of sarcomeres, and the calcium binds to troponin. Remember, the troponin-tropomyosin complex is the regulatory protein that controls contraction. When calcium binds to the troponin, it changes the shape of the troponin, and now myosin and actin can interact, and you shorten the cardiomyocyte. The sarcomeres contract, you squeeze on the blood. So those are the 10 steps, if you will, that does this. And it's a sodium gradient that is going to cause this depolarization. Now, once the signal stops, all right, so you get the burst of electrical current or the positive charge from the SA node, and all of this happens down to contraction, then we have to repolarize or remove the calcium. If the calcium stays in the cytoplasm, the cardiomyocyte stays contracted. So what happens here is at the same time, we have a couple different pumps working here. We have a calcium pump that's always pumping calcium back into the SR. So once depolarization stops and we repolarize the cell by opening potassium channels, calcium is going to flow into the SR by this calcium pump. There's also a sodium-calcium exchanger here. This is a antiporter that is run by the sodium-potassium pumps that are found in the cardiomyocyte. We allow calcium down its concentration gradient, and we pump calcium out into the extracellular fluid. This NCX is a sodium calcium exchanger. There are drugs that target this, right? That shut them down so that calcium's not removed and calcium stays in the cytoplasm when you take these drugs. And these drugs are taken, for example, with patients who have congestive heart failure. Their heart's not working as well. It doesn't contract as hard. All right. It's been damaged. And so it can't produce contraction on its own normally at a strong enough contraction rate that will produce enough blood movement. So what they do is they give you these drugs, which will slow down this exchanger, and that keeps calcium high in the cytoplasm. The more calcium you have in the cytoplasm, the harder the heart is going to contract with each beat because of the leftover calcium. So we have specific targets to these calcium exchangers that can be used by the pharmaceutical industry to help individuals with congestive heart failure, for example. So this calcium exchanger and this pump restore calcium levels to low so that the muscle can relax. And then you get another burst of electricity from the SA node, passes through the gap junction, opens sodium channels, depolarizes the cell, opens L-type calcium channels, which cause the opening of the ryanodine channels, which cause calcium spark and signal, and then contraction happens again. So that's how individual cardiomyocytes depolarize and contract, which is the major part of what the heart does, right? So this is kind of that conducting system I've talked about. This is the conducting system taken out of the heart so you can see it, how it's all kind of put together. Here's the SA node. Here's the AV node. Here are these internodal pathways. There's 1, 2, 3 of them that deliver the action potential directly to the AV node. There is an AV delay. That is the SA node. When it depolarizes the whole atrial network, which is a little bit slower than the internodal pathways. And so what that means is when the signal reaches the AV node takes a couple milliseconds, really about 100 milliseconds or so, before it's actually depolarizes. And that's because the proteins that receive that voltage from the internodal pathways are slightly different. And so they open a little bit slower than the SA node channels do. That causes the AV delay, which allows the atria to completely contract and eject their blood and relax before the ventricles start contracting. The internodal speed is much faster, so we got to have that AV node delay. And then the AV cells are not directly connected to the cardiomyocytes. They actually are connected to the bundle of His, which breaks down into bundle branches, which feed the left and right side of the hearts. They actually break down into Purkinje fibers, which are concentrated at the apex and move up. So the contraction starts at the apex and pushes the blood up to the blood vessels that leave the heart on its cranial or its superior side. As I mentioned, here are the heart rates of these different cells. All of these cells, the SA node cells, the AV node cells, and even the bundle branch cells can produce their own action potentials. The SA node controls everybody because it depolarizes the fastest, anywhere from 60 to 80 times a minute, where the AV valve by itself is only 50 beats per minute. So now you see why the atria and ventricles have to be coordinated through the internodal pathway, because the atria will be contracting really hard and the ventricle will be contracting every once in a while, like this, right? You want them to contract at the same time, like that, right? And that's what the AV internodal pathways do. They connect the SA and the AV nodes together and coordinate contraction. So that's kind of how the SA and AV nodes work, and then how the internodal pathways work, and the bundle branches and the Purkinje cells. Now, we've been talking about contractile cells and how contractile cells work. Now, you can measure the electrical output of the entire heart. All right, if you measure all of the electricity coming from the heart, we can do that at the surface of the body through what we call an EKG. All right, and an EKG looks a lot different. The peaks and waves look a lot different than what I've already showed you, and that's because it's the total output, electrical output of the heart. And this is what an EKG looks like. Here's a picture of the EKG here and showing you all of what are called intervals, waves, and segments. Waves are peaks that you see. They can be positive peaks or they can be negative peaks down here. The lines that are flat, those are called segments, and they run between waves. And then waves plus segments, they equal intervals. The names P, Q, R, S, and T, which you may have heard before, are the names of the waves. Now, an EKG is measured by electrodes, and those electrodes are placed on the surface of the body. Typically, if you're doing a 3-lead EKG, they're going to be placed on the wrist and one on the ankle. If you go into the hospital like I did and they do a 16-lead, they'll have 16 different electrodes all over your chest, some on your arms, some on your ankles that they use. And what they can do is they can use those leads and they can test lead 1 to lead 2, lead 2 to 16, lead 8 to 10, and so forth. And they can look at the electrical current through different areas of the heart so they can see if there's trouble somewhere within the heart itself. It gets really complicated when they start looking at things three-dimensionally and they use this EKG axis or this atrial axis to see where the heart is functioning normally and where it's not functioning normally. We're not going to do any of that. We're simply going to talk about what a normal EKG is and what each of these waves stand for. So that's on the next slide. So with an EKG, you'll get these small electrical currents. I got this neat animation up here, which gives you the EKG on the bottom and what's happening in the heart. And you can see here, if it will start over, right here when we get our P wave. What the P wave is the actual depolarization of the atria. That's the first wave. And then the complex QRS wave is the depolarization of the AV bundles and the ventricular network. And then the T wave is the repolarization of the ventricular mass. So P wave is atrial depolarization. The QRS wave is ventricular depolarization. And then the T wave is ventricular repolarization, where we bring it back to resting membrane potential. The atrial repolarization happens during the QRS wave. So you don't see it, but it's happening in that wave. The QRS wave masks it. So these waves, heights, sizes, how much space there is between the segments and the intervals, all can be utilized by a clinician to see if there's something going on with your heart. If your P wave is small or your T wave is tall or your segments between them are lengthened or shortened, then there's something going on mechanically with the heart. And then they can start looking into that kind of thing. EKGs are easy to do, and they're easily used by a diagnostic clinician to see if something is wrong electrically with the heart. It doesn't tell you anything about the mechanical aspect of the heart, just the electrical output of the heart. But it will allow you as a clinician to say, hey, we need to look into this because the heart's not performing well electrically. That affects the mechanical aspects of the heart. Okay, again, I throw this figure up here that shows you a comparison between skeletal muscle, contractile myocardial cells, and autorhythmic myocardial cells. So it's a little different than the one that has smooth muscle on here. It actually gives you the two major cell types of the myocardium, the middle layer there of the heart wall. What I want you to look at is membrane potential here. So this is resting membrane potential. Notice skeletal muscle is -70. Contractile cells are the most negative, -90. And then your autorhythmic cells, they really don't have resting membrane potential, right? They're very unstable, but they do reach rest at -60, but they creep immediately, right? Because of funny current. So what leads to threshold is net sodium channel opening in skeletal muscle via ACH, which is acetylcholine. So it's neurotransmitter driven by a nerve. Myocardial cells, it's depolarization via gap junctions. And with autorhythmic cells, it's these funny current channels that open and close, right? That cause depolarization. And what causes the rising phase of the action potential? once you reach threshold, is sodium in skeletal muscle and sodium entry in contractile myocardial cells and calcium entry in the autorhythmic cells, which are slightly different. Repolarization is opening of potassium channels, right? And opening of potassium channels. So potassium channels repolarize the cell, make it negative again. Hyperpolarization happens in skeletal muscle. No real hyperpolarization in the other two because of IF channels and the weird way the potassium channels work in the contractile cells. Duration of the action potential, very short in skeletal muscle, like I showed you earlier. Elongated, about 200 to 300 milliseconds in the contractile cells, and varies depending on what's going on within the autorhythmic cells. Refractory period, generally very brief, so you can stimulate skeletal muscle multiple times in a second. Long in cardiomyocytes because you want them to completely contract and relax before you can stimulate them again. And then it's not significant. It doesn't really affect autorhythmic cells at all because they're always creeping anyway. They never rest. So that's a really nice comparison across skeletal and the two types of cardiac myocytes that you see. All right, the next thing I like to talk about are kind of how we affect the heart and change its rhythm and all of that. But I did throw this slide in here because a lot of what we'll talk about here in a minute is mostly mammalian. And then at the end, we'll do a comparison between mammals and fish, just so that you have some, a comparative aspect here. This is what a typical four-chambered heart looks like. And this shows you blood flow through the heart, which you should know. So that you can make answer general questions and participate in general discussion on the exam. With the mammal heart, we have four chambers altogether, right? We have two atria on top, two ventricles on the bottom. The atria empty through valves. These are called atrioventricular valves. There's a bicuspid valve on the left side, which has two flaps, and a tricuspid valve on the right side that has three flaps. Two flaps on the left side because it's easier to control two flaps. The left side of the heart produces a much more force than the right side. And that's because the left side empties through the aorta and delivers blood out to systemic circulation where there are a lot more blood vessels. Whereas the right side of the heart ejects the same amount of blood as the left side. It just ejects it at a much lower pressure because it only has to send blood over here to the lungs, right? So heart and lungs are right next to each other. You don't need a lot of pressure to force blood through that part of the circulatory loop. Whereas the systemic circulation, I need high blood pressure to push it all the way through. The left pump is pushing oxygenated blood. The right pump is pushing deoxygenated blood. Blood flow, if you start in the left side of a drop of blood with the left atrium, when it contracts, will force blood through the bicuspid valve, filling the left ventricle. When the left ventricle contracts, it will force blood up because remember, contraction starts at the apex, goes up, closes the bicuspid valve and enters the aorta. The aorta then delivers blood to all of the arteries of the body. Those arteries break down into smaller and smaller arteries until they break down into the smallest arteries, which are called arterioles, right? Like we've already mentioned before. Arterioles drain into the smallest blood vessels we call capillaries. Capillaries drain into venules, right? Which are the smallest veins. These three vessels are called microcirculation. Exchange only happens in the capillaries, the smallest blood vessels. Once blood gets into the venules, it will drain into veins, and all of the veins will deliver blood back to the heart through the two vena cavas, the largest veins. They drain into the right atrium. The right atrium contracts, forcing the blood through the tricuspid valve and into the right ventricle. The right ventricle contracts, forcing blood up into the pulmonary trunk, which delivers blood to the right and left pulmonary arteries, which deliver blood to the lungs, where you pick up oxygen in the lung capillaries and return blood to the heart through the pulmonary veins, and the pulmonary veins drain into the left side of the heart. And so that's blood flow through a typical mammal heart. You can follow the arrows there on the figure, right? This shows you that blood flow, which we just went over. All right, so understanding the kind of the way blood flows through a normal mammalian heart will help us understand kind of the mechanical and physiological events that happen with each heartbeat. And so these are the mechanical events of the heart. Since we did the electrical events, right? We can then talk about what happens when we stimulate the heart with electricity. And so this is a cycle through a heartbeat, a single heartbeat. We can keep track of a number of things here. Notice in the circle here, we have when the heart is contracting and when it's relaxing, and that has a particular name. When the heart relaxes, it's called diastole. As you can see, I'm circling that word with my pointer. When the heart contracts, it's called systole. And then we obviously have atrial and ventricular diastole and systole. And then the S's here stand for the heart sounds, the lub-dub that you hear. If you put your head on somebody's chest and you listen to their heart, it'll be lub-dub, 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 right? Those are the heart sounds. Those heart sounds happen because of turbulence that happens underneath the valves. So the first heart sound happens here, and the second heart sound happens here. So talk about those in a second. But let's start up here in what's called late diastole. All right, so we got at late diastole, both chambers, your atria and ventricles, are both relaxed. They're not contracting at all. And the ventricles are filling passively. So what happens when the atria and ventricles are relaxed completely, they have the largest volume, and that draws blood into them. So the blood from the pulmonary veins and the blood from the vena cavas are filling the atria, which are now stretching, right? And that blood's heavy. And so by gravity, both valves open, the bicuspid and tricuspid, and blood actually starts spilling into the ventricles even before contraction happens of the atria. At the end of late diastole, we get next what's called atrial systole. So the SA node fires, the action potential spreads through the atrial networks. The atria then contract and eject the rest of the blood from the atria into the ventricles. So the ventricles are filling completely now. Basically at the end of ventricular rest, right before the ventricles contract, we have an end diastolic volume filling the ventricles of about 135 mils of blood. Each ventricle is holding the same amount of blood, about 135 mils at the end of atrial systole and at the end of ventricular diastole. End diastolic volume is the volume of blood in the ventricles at 135 mils. The action potential eventually gets to the AV node. The AV node, through the internodal pathway, right, has a delay, and then it fires, sending the action potential down the bundles of His and the bundle branches to the Purkinje fibers, and then the ventricles start to contract from the apex up. And as they contract and push the blood up, for a couple milliseconds, both valves close and blood is not being moved in either chamber. And this is called isovolumetric ventricular contraction. So the isovolumic means, sometimes you see volumetric, means that blood volume isn't changing at all because the valves are closed. The AV valves are closed, and also the valves of the pulmonary trunk and the valve of the aorta are closed. These are called semilunar valves. And so no blood is moving. As pressure builds up and in the ventricles, eventually the semilunar valves give. The pulmonary valve opens, semilunar valve opens, and the aortic semilunar valve opens, and then the ventricles eject blood into circulation, into pulmonary circulation and systemic circulation. And they eject about 65 to 70 mils. We'll say 70 mils in our picture here, because what's left over in the ventricles at the end of systolic contraction of the ventricles is about 65 mils. So we've ejected about that much. We can't empty the ventricles. It's just not possible. So 65 mils of blood remain in the ventricles at the end of ventricular contraction. As the ventricles begin to relax, what happens is the blood is going to drop back in the pulmonary trunk and in the aortic arch here. And that's going to close the semilunar valves. And for a couple milliseconds, all the valves are closed again. And this is called isovolumetric ventricular relaxation. And at that point, you get your second heart sound, which blood will pool around and be turbulent on on the side of the pulmonary trunk and the aortic arch where they've closed the semilunar valves. Gravity pulls them down. And so that gives you your second heart sound. Your first heart sound happened over here under S1 when the ventricle started to contract and the blood was pushed up. Remember, contraction starts at the apex, and that closes the AV valves and causes blood to swirl under there. And that gives you the lub part. So the lub dub are because of turbulence around the valves, both the first and second sound. So this closing of the semilunar valves gives you this few milliseconds where volume isn't changing in the ventricles. And that's called isovolumetric ventricular relaxation. Remember, number 3 was called isovolumetric ventricular contraction that closed all the valves. The atria will start to fill here, and eventually that will lead into late diastole of the ventricles where blood is so heavy on the AV valves that they open and start filling the ventricle because, again, and this cycle starts over. So those are all of the mechanical events of a single heartbeat. Now understanding that allows us to understand or start to talk about cardiac assessment. You can assess the heart and how much blood it's moving per beat and per minute. The ejection of about 60 to 70 mils per heartbeat is called stroke volume. The heart is typically in the mammal about 60% efficient as far as ejecting blood. That means you never can empty it. Some blood remains in the heart. We can increase or decrease stroke volume by increasing or decreasing either the heart rate or contraction, force of contraction. We can do increase in force of contraction by a mechanism that's called the Frank-Starling mechanism or the Frank-Starling law. And this is an intrinsically built mechanism into the heart. That is, if you stretch the wall of the heart more, the heart contracts immediately harder. And the reason for that is because your sarcomere length has an optimal length where contraction is the most efficient. So if you stretch the heart enough and you stretch it to where it hits the sac that surrounds it, that filling of the heart is the perfect length or the perfect stretch of the sarcomeres to get maximum contraction force. And so if you fill the heart to maximum, you get the hardest contraction. And the membrane around the heart that prevents overstretching of the sarcomeres is called the pericardium. And the pericardium is a dense, irregular connective tissue sac that the heart sits in that's non-stretchable to prevent the heart from filling too much and stretching the sarcomeres too much. Because if you stretch the sarcomeres too much, the overlap of actin and myosin is less and contraction will actually decrease. Contraction force will decrease. So greater stretch to the ventricles causes harder contraction, and then you increase stroke volume, the more blood you can eject. So think about it. When you're sitting listening to this lecture and you're kind of tired, right? Your heart rate's going to be basically not contracting as hard, right? Your heart, you can even listen to it or feel it, and the heart contraction, the force of contraction is less because the amount of blood being returned to the ventricles is less because your body's not working, right? Now, if you get up and you start moving around, your legs start contracting, right? The skeletal muscle in the legs, that forces blood back into the veins. More blood comes back to the ventricle. The ventricle starts stretching all of a sudden. More stretch, harder contraction. Stroke volume goes up to increase blood flow to those skeletal muscles. So it's a nice built-in intrinsic mechanism so that the heart can match input. The amount of blood coming in, they can match with the output of blood leaving because of stretch. And that's called Frank-Starling's mechanism. Now, cardiac output is stroke volume times heart rate, and that gives you an idea of how much blood is leaving. And let's say normally 70 mils leave per beat, and let's say the normal person has 80 beats per minute. If you multiply 70 times 80, it's like over 5 liters of fluid are leaving the heart every minute. And so the heart is very effective in pumping blood. And it's very effective in pumping blood under very high pressure. During systole of the left ventricle, 120 millimeters of mercury are moved. And that's how much pressure is produced, so it's a lot of pressure. There are a number of intrinsic factors that can alter cardiac output and stroke volume. These include the intrinsic factors like stretch, nervous control. The parasympathetic and sympathetic nervous systems can cause increases or decreases in heart rate and contraction force. And the endocrine system, hormones can do this also. Now, exercise can have a major effect on heart performance. If you exercise regularly, let's say you run 5 miles a day, versus somebody that sits on the couch and eats ice cream all day long. Both of those people, the person that's kind of sitting on the couch and doing nothing and the person who exercises regularly will move the same amount of blood per minute, about 5.25 liters of blood per minute. That's the cardiac output. The difference will be in their heart rate. So a person who exercises and works the heart, which is a muscle, will have a more efficient heart. It will beat less per minute. That's 44 beats per minute. Versus the person who doesn't work the heart that has a heart rate of 75 beats. What this means is the person with the lower heart rate has more time between contractions to fill the ventricles. The more fill you have, the harder the contraction, right? Frank Starling's mechanism. They eject more blood. Their heart is more efficient because they work that muscle. This becomes apparent when the person on the couch gets up and tries to chase the person who runs all the time. And they double their contraction rate. And this is the double resting heart rate is 87.5 when this person starts running, so which is really close to 75 beats per minute. Whereas the person who doesn't exercise, the heart rate's going to go above 150 and they're going to tire really quickly. So this person can outrun you really easily because they obviously work the heart all the time. So exercise, cardiovascular exercise in particular, is really good for your heart. Right? Your heart is a muscle, you need to work it. So Starling's law is what we've been talking about. And Starling stated that preload, what's called preload, can increase contractile force. Preload is how much stretch there is in the ventricles. The more blood that's returned to the heart, the more stretch you get, the harder the ventricles contract. That's called preload. So preload fills the ventricles and stretches the muscle to optimal length, and you get great contraction force from that. There are many factors that can increase stretch. One of the major ones is venous return. So how can we increase venous return to get blood back to the heart, to stretch it more, to cause it to contract harder? And you can increase heart rate to do that. You can increase force of contractility. You can stretch the heart more. You can release hormones. There are a number of ways to do this. And I will show you those on another slide, which is the slide right here, right? The next one. So the main controllers of cardiac output are based on stretch. Increased stretch increases stroke volume, increases cardiac output. So how can we increase stretch? Increase venous return. Well, how can we increase venous return? Well, we do this in a couple ways. Some of it's already built in. Veins, most of the veins of the limbs in particular, but in the body, are going to be found between antagonist groups of skeletal muscle. So that if you move your legs or your arms, what you're doing is also squeezing veins, which is returning blood back to the heart, which increases stretch and can increase force of contractility. Veins also have valves in them to prevent backflow, to keep blood moving efficiently back towards the heart. Smooth muscle, there are smooth muscle in the veins, and you can stimulate them through nervous control to contract and force blood back to the heart faster or more efficiently, again, increasing venous return. You can increase blood volume through the kidneys using hormones, which we've already talked about, or through the thirst response, right? These things can increase blood volume. That's a more long-term goal of the body. And then sphincters, capillary beds throughout the body have sphincters on them that can close down and redirect blood back to systemic circulation to increase blood volume and then increase venous return. These sphincters are very efficient. They are basically control blood flow to capillary beds based on the activity of the tissue. So that's really nice also. So these are all kinds of built-in intrinsic factors the body has to increase or make sure venous return is efficient enough to run your body processes. Okay, nervous control. As I mentioned, there are two systems, the parasympathetic and sympathetic nervous systems that control heart rate. Parasympathetic nervous system slows the heart down. The sympathetic nervous system increases heart rate and increases force of contractility. The sympathetic nerves are going to innervate or send nerves directly to the SA node and to the contractile cardiomyocytes. The sympathetic nervous system only sends nerves to the SA node and AV nodes, so it doesn't really affect force of contraction. So the sympathetic nerves are going to release neurotransmitters that increase the amount of calcium released, and that's called ionotropic mechanism. An ionotropic mechanism increases calcium concentration inside of the muscle cytoplasm, which will increase the amount of actin and myosin that can interact, and force of contraction will go up. Also, the sympathetic nervous system is going to open channels that are going to increase heart rate. Parasympathetic system is going to open channels that slow heart rate, or sometimes close channels, right, that can slow heart rate. And so the next slide, I will show you kind of how these two systems work on the heart muscle itself. And so these two slides do that. The parasympathetic nerves, they release ACH, which is acetylcholine. So that's one of the neurotransmitters. The sympathetic nervous system releases NE, which is norepinephrine, right? Norepinephrine. So norepinephrine is spelled up here for you, or epinephrine. We can really treat them the same. So notice parasympathetic nerves, they bind to specific receptors on the autorhythmic cells that are called muscarinic receptors. That's one type of acetylcholine receptor. Muscarinic is named because it binds the mushroom toxin called muscarine. That's where the name comes from. That's how they were discovered, and they're called muscarinic receptors. And they're found in the autorhythmic cells. And what a muscarinic receptor, once it's bound to acetylcholine, what happens is you get an increased efflux of potassium and a decrease of calcium influx in the autorhythmic cells. That mechanism is located up here, okay? So make sure you guys are paying attention to this. So this side here represents the parasympathetic, right? And so notice acetylcholine comes in, binds to the muscarinic receptor. The muscarinic receptor is a G-coupled receptor. And the G protein, once activated because of the change in the shape of the receptor, the alpha subunit comes off and will bind to two different channels. It binds directly to the T-type calcium channels, closing them. Remember, the T-type calcium channels are responsible for finishing off the funny current, right? And so typically what would happen is you get something like this, right? With the parasympathetic nerves, if I affect these channels and close most of them, right there, then I'm going to flatten this and my heart rate's going to go down. It's going to look like this, right? And so if each depolarization causes a contraction, obviously this one is going to cause less contractions, right? Or slow heart rate. And so closing the T-type channels is going to do that. It's going to flatten my funny current, take longer to reach threshold. And it also opens more of those potassium channels, kind of hyperpolarizing the cell, making it harder to reach threshold. So it's going to slow heart rate. So the hyperpolarized cell, rate of depolarization goes down, heart rate goes down, right? Parasympathetic nerves decrease heart rate, decrease conduction rate, decrease contractility, especially since we're affecting calcium, and that's what acetylcholine does. Now, epinephrine, on the other hand, which is over here, right? What it does is it binds to what's called a beta receptor. The beta receptor, there are two types of epinephrine receptor, beta and alpha. In the heart, we have beta receptors. And that's going to increase sodium and calcium influx. That increases the rate of depolarization and increases heart rate. And so this is a G-coupled adenylate cyclase receptor, the beta receptor. So norepinephrine binds to it, activates the G protein. The alpha subunit is released. It binds to adenylate cyclase and takes ATP and forms CAMP, or cyclic AMP. CAMP activates protein kinase. And the protein kinase here is actually protein kinase A. Protein kinase A phosphorylates funny current channels. The sodium channels opens more of them. More sodium flows in. You reach depolarization faster. All right. So now you start doing this, right? Putting the depolarizations really close together, like that. You contract. It increases force of contraction. You open up more T-type calcium channels, more calcium floods, again, increasing depolarization rate and also increasing the force of contraction. The more calcium you have, the harder you can contract. So that's kind of the mechanism behind each. I will do a lab where I send you a video of a heart lab where I opened up a frog and stimulated the frog with different hormones like epinephrine and different neurotransmitters like acetylcholine, and you'll be able to see the heart speed up and slow down. It's very nice. It'll go along really nicely with this particular lecture. So I'll put those online probably in 2 weeks, right? Once I get back next week from traveling with my family. So we have these ways to speed up and slow down heart rate, right? But what mechanism out there or what parts of the body kind of monitor heart rate, right? And so we're going to have negative feedback loops that can stimulate the sympathetic and parasympathetic systems. And that's what this shows here. This shows you that the heart and some of the major blood vessels have a specialized receptor called a baroreceptor. What a baroreceptor does is it senses stretch, right? When you pull on the receptor, you stimulate it. And so there's normal stretch with heart rate that the baroreceptor picks up. If you get increased stretch or decreased stretch, right, then you can stimulate these baroreceptors, which will then stimulate the sympathetic and parasympathetic nervous systems. The sympathetic and parasympathetic nervous systems are going to be found in the medulla, part of the medulla, which is the upper part of the spinal cord, or the brainstem, if you will. And it's set up with reflexes. So the baroreceptor sends sensory information, as you can see here, into the cardiovascular center, if you will, of the medulla. And what happens here is if atrial pressure or arterial pressure drops or falls, the baroreceptors will sense that with less stretch, and then they will send motor output through the sympathetic nervous system. And the sympathetic nerves will increase heart rate and cardiac output by releasing epinephrine to the SA node into the cardiomyocytes. If blood pressure is too high and you get too much stretch, the baroreceptors will stimulate the nerves in the brainstem here of the medulla and send output through the parasympathetic nerves to slow heart rate and decrease blood pressure and cardiac output by releasing acetylcholine to the SA node and the AB nodes. And so that these baroreceptors and the cardiovascular center in the medulla oblongata of the brainstem are what kind of control extrinsic heart rate, right? Increasing or decreasing heart rate when needed. Endocrine control. We can release epinephrine into the blood supply too through the adrenal glands. The adrenal medulla in particular, can induce the flight or fight response. So if somebody comes up and grabs you, or you pull out in front of a car and slam on your brakes, your heart rate goes up and you're energized. What happens there with that release of sugar into the bloodstream and the increase in heart rate is the adrenal glands respond by releasing epinephrine, which travels again to the heart and increases contractility and heart rate. You can also slow the heart down through a hormone that's released by the atria, believe it or not. When the atria are stretching too much, they're working too hard, they will release ANP, atrionatriuretic peptide. And what natriuretic means is it works on the urinary system and the absorption of sodium and chloride. And so what will happen here is ANP will cause an increase in volume of the urine. Right, it'll increase reabsorption of sodium chloride into the filtrate. And if I do that, more water stays in the filtrate, right? And if more water stays in the filtrate, then I urinate more. That will decrease blood volume and decrease stretch on the heart until it comes back to normal, and then blood volume is back to normal. So AMP is kind of antagonist to aldosterone and ADH. Okay, the last thing we'll cover here is I showed you a mammal heart. I also want to show you a fish heart. So fish hearts are a lot less efficient than tetrapod hearts, and there are only two major chambers. There's one atrium and one ventricle, all right? So here's the ventricle here. Here's the atrium up here, right? A lot of fish have what's called a caudal sinus or a caudal heart. The biggest problem with fish is when they swim forward, all the blood tends to pull back into the tail. That's just the way things go as far as the way gravity works in water. And so blood tends to pull back here when fish and sharks are swimming. And so a lot of sharks in particular, or larger fish, have a caudal heart, which is a contractile blood vessel that also contracts, that squeezes the blood forward. Most of the veins are located in the middle of the body so that the contraction of the body doesn't push all of the blood, right? If you're pushed up against the body wall, most of the muscle in the fish is found in the body wall, would push all of the blood to the tail. So this helps with not pushing so much blood back, and venous return is all in the middle so that it's more efficient to get it back to the heart. Atrial filling works with a vacuum. It actually stretches. Body movements stretch the atrium when the fish is moving like this. And that stretches the atrium, which suctions or pulls blood back. And caudal hearts help also. So that's pretty interesting with fish. But a fish heart is pretty simple, and so is the circulatory system. So the heart's set up like this. There's an atrium and a ventricle. The ventricle is very spongy. That is, a lot of blood stays in the ventricle because there's not a very good coronary blood supply to the heart. So the heart kind of has to feed itself oxygen. And it does that through the sponginess here. But blood does flow through the ventricles, which is very strong. It contracts and pushes blood out into the first part of the aorta, which is called the bulbous arteriosus. And then that will deliver blood to the gills. The gills pick up oxygen, send it to the head, and then venous return from the body sends it back to the atrium and ventricle. Ventricle contracts, pushing it back to the gills. You have a single loop here, right, of blood vessels. The systemic capillaries are in series with the heart. And so that's how a fish heart works. So fish heart's a little different than the mammalian hearts and even the amphibian heart.